Thank you very much, Dan. And for the final presentation before we have discussion, and we'll have plenty of time for discussion, it's my pleasure to invite Katrin Rupalla, the head of regulatory oncology at Bristol-Myers Squibb. We can set the clock to 20 minutes for her, too.
Thank you very much for the invitation to speak. I'm speaking here on behalf of one of my colleagues who told me on Friday I have to be here on Monday. But I will be focusing today as the head of oncology regulatory at BMS, mainly on the examples we have already with 3 approvals of IO-IO combination in the United States. I will talk about our experience, what were the regulatory challenges, and also what's coming next in terms of considerations. I have to tell you, I have moved to the US 3 months ago. I was the head of development in Shanghai, so dealing with 700,000 lung cancer patients per year and 600,000 deaths for lung cancer per year. So it's a quite different magnitude of unmet medical need, but a society that's had no access to PD-1s at this point. So it's for me very interesting now to actually move to a very different aspects of development and combination, which is the next era. I have the following disclosures. Of course, I'm employed by Bristol-Myers Squibb, and I have shares of Bristol-Myers Squibb. I want to focus on my presentation a little bit about the BMS strategy with combinations, then the opportunities and challenges we see with the development of combinations. I will focus a main part on the contribution of component with what was touched on by Dr. McKee as well as by my previous speaker. And then also consideration for drug development from a regulatory perspective. Why are we doing this? And I think this is because of the promise of immunotherapies to treat cancer. And I think the most exciting part is the tail end of the curve. So what we are really trying to achieve is to lift up the end of the curve and really try to have long-term survival. I think if you talk about combinations, when you look at also nivolumab, ipilimumab data or nivolumab monotherapy data in lung cancer, especially in the non-squamous population, second line, you clearly see that there's an overlap of the curves early on in the treatment, but that the separation of the curves only happens after 6 months, PFS curves, the separation or survival curves. So the goal or the dream is to actually do both, lift the tail end of the curve with combinations, but also with the combinations try to have an early benefit in the treatment with immune therapies. So that's why combination is so attractive for studying further. Based on our track record in developing treatments based on immunotherapy concept, actually the main focus of our pipeline is concentrating. Our main targets will be in the T effector cells. So really targeting immune cells, immune stimulation, inflammation in the tumor. We have less developments on the targets, direct targets of tumor cells. So that's also why we have maybe a little bit of a different view on contribution of component, because now we are talking about a mechanism of action that has new targets, has never been shown, and is combining two agents with these new targets together, rather than established chemotherapy or combining it with targeted therapies like TKIs. In terms of our experience with late-stage development and approvals, I will be focusing on the challenge of combinations. The challenge, I believe, is mainly with new combinations which have a new mechanism of action or are less established, like my previous speaker was saying, Avastin in bevacizumab in HCC. Less established, indications for or less established compounds in certain indications. I personally believe we will not go back in time and ask the different components of chemotherapy, like pemetrexed, for example. So case by case, we also have to look case by case development with the tailored study designs, and I will give you the 3 examples of the recent approvals for ipi and nivolumab to just demonstrate one size fits all is not possible in this situation. It depends on the data, it depends on the scientific rationale, and I will talk about the 3 specific examples here, which is the combination of ipilimumab and nivolumab in first-line metastatic melanoma, then in advanced renal cell carcinoma, and then the most recent one, which was just last week in the combination in third-line CRC MSI high. So, of course, everything needs to start with a strong scientific rationale. And the rationale for combining nivolumab with ipilimumab was actually touched on a couple of times today already, is in terms of ipilimumab is more considered to prime T cells or have an effect of T cell expansion, so you increase the number of T cells. And then if you combine it with a PD-1 inhibitor, you actually take the brake off the T cells in order to recognize the tumor cell. So the strong scientific rationale needs to be the foundation of any combination that's being studied. Then we come to the data. And I'm also telling a little bit about RCC because it's a quite interesting observation also, because sometimes it's also not possible to go back in time. So when you look at the phase 1/2 data that we generated with the ipilimumab and nivolumab combination, in melanoma we had 60 to 40%, so 20% difference in ORR. When I come to RCC, it's a very interesting case, is if you look at the combination, it was 40% ORR, but when we looked at monotherapy, it was only 20%. And then if you look at sunitinib, first-line response rate is about 26%. At this point, you would not take a monotherapy forward if you compare it to a standard of care, which has already an increased ORR. And then in CRC, for example, the difference is even bigger in terms of response rate, almost 30% between the combination and the monotherapy. Then the key challenges for the development of the combination is not only increased efficacy, but it also comes usually with an increased toxicity. Also what we have talked about the whole day today is about fragmentation of patient population by biomarker and who benefits the most from a combination. We have different biomarkers now being evaluated, PD-L1, of course, tumor mutational burden, inflammation, immune signatures, all the biomarkers we spoke about today. And then the contribution of component, like the previous speaker touched on, is single agent contribution of nivolumab and ipilimumab. And what is the evidence that we need? Let's start with the example of first-line melanoma and the approval in the US. So the first approval for the ipi-nivo combination was in first-line melanoma based on a phase 2 study. Very simple design. Ipilimumab was the standard of care. So that was compound B. And then we added compound A, which was nivolumab. So we had a very straightforward design, AB versus B. So it's easy to establish what the contribution of A was in the regimen. But that left us with a question. What is the activity of a monotherapy? Which means what is the activity of nivolumab monotherapy comparing to the combination as well? So that meant actually we set up another study, which was a phase 3 3-arm study that now could also look into the contribution of A alone and how it relates to the combination at the end of A and B. So this is Checkmate 067. It's a 3-arm study. The design was AB versus A versus B. Also very straightforward, 2 monocomponents, B and A, and then the combination. So very, very straightforward in terms of establishing contribution of component. If you look at the safety results of Checkmate 067, which was nivolumab monotherapy, ipilimumab as monotherapy as standard of care, and then the combination. And I have to say this is a regimen using 3 milligram ipilimumab and only 1 milligram nivolumab. So from a design perspective, it's also very easy to delineate the added toxicity to the monotherapy of each of the components, right? And here you can clearly see that in terms of toxicity, ipilimumab 3 milligram per kilogram really added to the toxicity of nivolumab. So also in terms of safety profile, you could really delineate in this design what is the added toxicity compared to the monotherapy. The story is a little bit more complex if we go to first-line RCC. So that was a design, ipilimumab plus nivolumab versus the standard of care. So this is A plus B versus standard of care, which is sunitinib, right? So how do you establish a contribution of A and B in this component, in this regimen versus the standard of care? So the rationale for designing the study was that we knew that ipilimumab had minimal activity in RCC as a monotherapy. When we looked at the combination nivolumab plus ipilimumab, we saw that we had a doubling of the ORR compared to nivolumab alone. And then we had a standard of care, which was a TKI. So that was the rationale for the design. And I want to come back is to say, right, is sunitinib has a response rate of 26% in this study. If you look at the response rate of monotherapy nivolumab that we established in phase 2, and I just presented, was 20%. So nivolumab monotherapy, 20%. Ipilimumab, we knew almost no activity in RCC. And when we combined it, we had a response rate of 40%. So at this point, right, we needed to make a decision. We cannot study nivolumab monotherapy because compared to sunitinib, the ORR was already lower, and we knew ipilimumab monotherapy is almost no activity. So that was the only design that we could take forward into phase 3. Then another design, which is again looking at the contribution of component, is we had in colorectal cancer, we have an approval for monotherapy in MSI high, and that's based on the Checkmate study 142, which contained another cohort, which was the combination ipilimumab, nivolumab, but in a non-randomized fashion. It's a third-line setting, 2 parallel cohorts, one with the monotherapy, one with the combination. This time we studied nivolumab 3 milligram and ipilimumab 1 milligram, and I will highlight this in the safety results of the study. And if you look at the response rates for the 2 cohorts, a clear differentiation. If you look at the response rate in the monotherapy cohort, it is about 30%. If you look into the ipilimumab and nivolumab cohort, it's about 50%. So a 20% difference in the overall response rate. And then also if you look at the complete response and then also the proportion of response, there is a clear difference with the combination. But that leads to another question, right, is we are having discussions with the FDA is, do we need these data in a third-line setting from parallel cohorts, or do they need to be randomized cohorts, right? In this case, this is a non-randomized cohort, and they have been slightly recruiting in parallel, but not fully overlapping in parallel recruitment. If you look now at the safety data, here nivolumab 3 plus ipi1, it's much, you see that almost the combination is almost similar in terms of safety compared to the monotherapy, right? So it does not always mean that if you add something on, you really change or have edited or make a huge difference in the safety profile by adding toxicity. It also depends on what the doses is of the different combination partners. So if you here compare the grade 3, for example, it's almost no difference between the combination, but this is with low dose ipilimumab. And then for general consideration based on our experience is when were the data generated and how reliable? So early data may look significantly better. We have a very good example here, is epocadostat and pembrolizumab combination. If you look at the phase 1 data, outstanding. So we can have, it's an overestimation of the treatment effect size of the combination. But in this case, in renal cell carcinoma, for example, we had a 20% monotherapy response rate with nivolumab, whereas we believe today that response rate is most probably higher. So we may want to, if we would design the study today in RCC, most probably we would include a monotherapy arm for nivolumab. But at the time we made the decision, we did not have the evidence. So it's also very difficult to go back in time once you have made a decision on a certain data set if you study the contribution of component. The other question I just touched on is, and also the consideration Dr. McKee presented, is ethical considerations. It's not so straightforward. Here are less tangible decision-making possible. Can we really include a monotherapy arm if we have clearly seen a difference with the combination arms in terms of efficacy, especially in mid and later stage trials? Different population, what we see now, right, is if you look at the CheckMate 026 results that were presented this morning and a finding of a new biomarker that is TMB enriched population, how can you go back in time also with contribution of component when you had an unselected patient population? You even don't have TMB data left from monotherapy studies. How do you then establish the contribution of component? Do you have to go back to the drawing board and study again the monotherapy in the enriched population before you move into the combination? It's a multitude of questions that you need to ask yourself, which data, what population can you do extrapolation, and what is feasible in terms of conducting the study. Also, I would like to make one comment about master protocols because it was touched on in the morning and also touched just now by the previous speaker, because this is a session on regulatory challenges of combinations. I just wanted to say that we have since 3 years a master protocol running, very similar to the Morpheus study. But the challenge was to get the study up and running. We met in Europe, we met 18 different health authorities just to discuss how we get the study approved and then how we actually can amend the study and introduce new combinations. So it's not a globally accepted concept. In the US and Canada, it was much, much easier. But outside of the US, the master protocols are actually a little bit more challenging in terms of administrative hurdles. Again, also multi-cohort trials are not always acceptable. We have no approval of CRC MSI high in Europe. And also, non-randomized studies, single-arm cohort studies are not always everywhere acceptable. Then also, clearly, we see differences how the EMA approaches contribution of component questions compared to the FDA. I would say in 80% it's similar, but in 20% it's just a different angle of where the authorities come from. Sometimes it's the question on nivolumab monotherapy. In the other cases, it's the question of ipilimumab monotherapy. So looking at it from a different, which component is more important for asking the question from a regulatory perspective. So we see also some differences here. Then what endpoints can be used, especially in phase 2 studies? Is it sufficient to show ORR and duration of response to select the combination over the monotherapy for phase 3? And then, of course, it's very important to understand what are the data, the clinical evidence, the existing data from other companies. And then actually for us, the contribution of component is becoming a major discussion point during the design of development programs because it is part of the regulatory assessment. And therefore, this is something we really have to carefully plan for in order to obtain approval. With that, I want to thank you very much for the attention and I'm standing in between the dinner now and the reception. So thank you so much for your attention.